Hello and welcome to this webinar titled Chemical and Physical Transformation Monitoring by Raman and IR Spectroscopies in Pharmaceutical Development, hosted by Biopharma Asia magazine and presented by John Wesley, Senior Principal Scientist at Bristol-Myers Squibb, followed by Utility of Raman Spectroscopy in Continuous Processing of Liquids and Solids, presented by Karen Esmonde-White, Senior Marcom Specialist at Kaiser Optical Systems, Inc. My name is Stephen Edwards, and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live Q&A session at the end of both presentations. Audience members can send their questions in at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now, please allow me to introduce our first presenter. Dr. John Wesley Phd is a Senior Principal Scientist at Bristol-Myers Squibb Company. He leads a vibrational spectroscopy group who are focused on developing non-invasive, sustainable methods designed to enhance productivity and generate process knowledge. The spectroscopy-based methods have been deployed in areas ranging from early scale-up to manufacturing environments at various BMS sites. The developed methods encompass both small molecules and biologics. In addition, the group provides hands-on training and oversees open-access spectrometers for chemists and engineers. John is currently the marketing chair for the Federation of Analytical Chemistry and Spectroscopy Societies and Pharmaceutical and Biopharmaceutical Section Co-Chair for SCIEX 2019. I will now be handing over to our first presenter. Welcome, Dr. Wasylyk.
Thank you very much for the introduction, and we'll begin the presentation. The first series of slides, I'm going to be going over some general aspects of spectroscopy and why we use them, and some quick examples of items that I think are very relevant when you're in the pharmaceutical or chemical industry. Our capabilities are, we focus the talk today around Raman, both inline and offline, and FTIR. And you can see from the utility in our very first slide, we do both inline and offline in both instances, but we also add a number of other advantages in the case of Raman spectroscopy, where we're doing some very nice work with solvent exchanges and reagent analysis. We will do reagent analysis with both FTIR and Raman, and we do a lot of time course studies with Raman spectroscopy. Now, the next slide. Our capabilities are listed here, and I want to point out some of the aspects that we deal with are safety. So when we're doing an inline or offline assay, we're going to, in the bulk of the cases, avoid any kind of sample preparation or exposure to chemicals, and I'll have examples with that. I also want to stress at the bottom speed and quality. Inline or offline are much more rapid if you can do it non-invasively without having to have any extensive sample preparation. In the middle there, by doing inline analysis, especially, we can save on solvent waste, preparation, generation of additional waste. If we could put a probe in a vessel or shoot through a vessel with a Raman signal, for example, I take that as being an advantage towards green chemistry or sustainability. And that's one of the big things we're trying to push. Safety first, speed and quality, and also greenness. When I mention shooting into a vial, this is one of the tools of our trade. We take the Raman spectrometer. It has the laser goes through this microscope objective into the sample, and so non-invasively we can quantitate or qualitate the amount of material in the jar and do our analysis. This was in conjunction with Kieran O'Connor in Swords, for example, one of our collaborators in Ireland. And if you look here, he took some time to look at how long it takes to run a GC sample for a simple solvent exchange. And most chemical reactions have some sort of solvent exchange as you're going from step to step. And they have to do opening standards, closing standards, run the sample in duplicate. The time adds up. with the Raman spectrometer, we tend to run our assays in about 2-minute time scale, and we save a lot of time, so it enhances our productivity. And that's just for offline analysis, as I showed you shooting through a jar. We also pick instruments, whether it's FTIR or Raman or near-IR, that are ATEX rated, so we could use those in various countries, and of course UL rated for applications in the United States. And here's an example of an instrument on the left-hand side that's a rated instrument to work in an explosive environment, and also on the right hand is a probe with a large orange conduit, and the fiber optic line is attached to the probe that goes into the reactor or a sampling loop. This just demonstrates one of the tools that we have used extensively. That's Dynacam Modeling. Now with that modeling software, there's a number of them available, we could actually follow a distillation. And in this particular case, they start to distill, then they feed in isopropyl acetate at a constant volume, and they want to eliminate the acetonitrile. It needs to get below 5%. They also need to eliminate water and take that as low as possible. And you can see from, in this particular case, we can follow that distillation in real time. So in a matter of minutes, we know exactly where we are. In this particular case, we use the distillate line as opposed to putting the probe in the reactor. The reason being there is that what's coming out of your distillate line can be related to what's in your tank in real time, but also by modeling it, but also gives you the power to be able to use this model that you built on other distillations if you're involved in isopropyl acetate and acetonitrile. So every time you see that kind of distillation, we already have a model for it, so we can apply that and save a lot of time, and that enhances our productivity. Of course, you may question, well, how about Raman? You need to actually verify that it works in all cases or in particular cases. And here, with the forming of a shift base, we're able to monitor and pull samples, run HPLC, do the quantitative analysis, and we'll build a model from this data. This is going to be our primary method of analysis, and then we will develop a Raman model in this particular case and follow the reaction progression. So it works out it's advantageous to us. One of the things that I had mentioned in the earlier slide was this data calibration and then transfer of a method, especially if it's going to go into a manufacturing site and it's going to be filed. So we'll go to an external site or external manufacturing site, and we will co-validate. And it's something we've worked on because it's a lot easier to do a validation and divide the work. It's a lot easier than transferring, let's say, an HPLC method where it has to be revalidated in the new site with column. The tubing can change. Hopefully they have the exact same instrument, the flow, the sensitivity of the diode array detector or fixed wavelength detector, whatever they're using, or refractive index. In this particular case, with the instruments and the models we built, we're able to transfer the models directly from instrument to instrument, and we're able to share the spectral data, so we build robustness. So we'll run it on a few instruments here in our site, and when it goes elsewhere, they'll also run, and we can add that data to the same chemometric model and build robustness. And that also takes care of various types of reagent variabilities of what you purchase. That I'm going to touch on right away. Here's a quick example. Typical centrifugation washing of an active pharmaceutical ingredient, an API. By being able to put a probe in your solution or in the effluent or what's coming out from your flushing, you're able to monitor the washing process. So you're going to remove solvent A, take it to solvent B, and then start your drying process. And so this is one way that we can actually cut down on our waste. This adds to our sustainability or greenness approach. So instead of flushing for 9.5 hours, we can call this at 5 hours. This also gives you the ability to look at it in terms of volume sense. Do I have to do 5 volumes or 4 volumes? Some cases you may have to use 5 volumes of solvent for an exchange. In other cases, 1.25 may be sufficient, and you want to know that up front and then confirm that in the manufacturing site. Again, adding to the sustainability of using inline spectroscopy. I did want to touch on this because it's something that has really become very important for us, and that is using Raman and FTIR to look at the reagents which we use in our chemical reactions. This is just a couple of quick examples I'm going to breeze through. And you can see here just the simple FTIR spectra of sodium triacetoxy borohydrate. And there's a couple of lines in here, spectral bands, which are listed in green. They're the color green. Those were quality material. The other ones were also purchased lots which did not behave properly. In other words, the reaction did not go to completion. There were issues with the quality of the material, side products, etc. So what that shows you here is we can take a quick scan using FTIR and look at the quality of the material. Same thing here with Raman, and in this particular case, it was a stability study. And this was done in our lab, just taking the stab reagent, exposing it to lab air, and letting it run for an overnight period. And you could see the decomposition of it over time as it sat at room temperature exposed to laboratory air. What that is telling us is that if you buy your solvent in bulk, you want to make sure that it's purged. You are putting an overlay on it, let's say with argon. You don't want to have it overexposed to the air because you're going to be breaking that reagent down, and that's going to impact your reactions. And this is just a decay of the stab that we can generate over that 24-hour period. Now, this is also, we extended this to take a look at 13 different lots of this stab reagent, 13 different lots, and we compared that with the certificate of analysis from the various vendors. What you can see here is the best material based on the Raman was number 12. Number 11, there was very little of the stab remaining. So what we had received was very, very poor quality. So when you go to run your reaction, it will not go to completion. Matter of fact, it doesn't go anywhere. We have another example of that coming up. So you can see there's a lot of variability in purchase lots. So it's something that we try to tell all our chemists that if the reaction doesn't work, it could be your reagents. And let's take a look at it. This is another reagent that we recently looked at. We knew that it was going to go into a reaction and being fed in slowly. In that reaction, there will be a small amount of ammonium hydroxide. It's in methanol, water mixture. It's kept at room temperature under agitation. And this is how it's going to proceed in a reaction. So we just did a time course study using Raman to see how long before it decomposes. Because if they do a very slow feed, let's say it happens to be a 24-hour reaction, but if they also feed it in slowly, how much of it is going to decompose before it actually is able to react? And so you heat up your feeding system to put in your chlorination reagent. In this case, it was N-chlorosuccinimide. Now, I'm going to shift gears and go back towards the main topics here, and that has to do with the quality of following the reaction or the forms that you have to maintain during pharmaceutical development. In this particular case, I'm just going to show you some FTIR work and then Raman and back to FTIR. This happens to be a very slow mixing rate of chlorinating reagent that we used in a single addition. So we could see the chlorination reagent being added, a slight drop in it over time, and a product being formed. So we can follow this by putting the probe in a vessel using FTIR. In this particular case, the material crystallizes out. So it's not a complete saturation, or you're monitoring the saturation of what's being produced. You're actually getting a higher yield from it. This is a study which the chemists want to look at adding the chlorinating reagent, in this particular case, over a period of time in shots, in aliquots. And this way we can look at the various intermediates also being formed. So we can follow it, follow when the temperature goes up, and then during that process see it kick off and actually doing what it's supposed to do, chlorinating. This is also in the same kind of project where we're adding an acetylene, a cyclopropyl acetylene, and we can see what the initial addition of the cyclopropyl acetylene is added. We can see the triethylamine that is also being added, and that is going through the reaction process, so we could follow the progression and decide at what point during this reaction we can actually call it or stop the reaction. Initially, many times reactions tend to run overnight or throughout the day. By doing the simple aspect of putting a probe in a vessel, you can follow the kinetics of it and determine, yes, my reaction can be done in 6 hours or 4.5 hours. So it gives you a lot of the kinetic profile, and that's important for us in both final product and also intermediates. One of the aspects I'm going to touch on now is using a Raman spectrometer to look at a slurry-to-slurry form conversion. So this is a slurry, and there's both a kinetic and a thermodynamic product. Our thermodynamic product happened to be labeled or identified as N3. So our goal here is, can we monitor this during this conversion, save time, efficiency, and make sure it's going to completion? And of course, we'd like to be able to replace some of the more extensive, expensive X-ray analysis, for example, because it does take time to get those results. We want to get this in the plant, have it manufactured, and move on to the next step. So what you can see here is this N3 form. We didn't see good results with different types of instruments. We did see with Raman, for example, a slight change in a spectral band, and it really appears as just a shoulder at 1622. What this is going to show you is that we could still develop a method based on just that shoulder for the form conversion, going from the kinetic product to the thermodynamic product, and we can follow that. Now, typically in this particular case, we actually add a seed to it. We add some N3, and that helps that conversion move along a little faster. Which was done, approaches were putting a probe in the vessel in a small reactor, or using a non-contact objective and shooting into the vial and have that vial stirring on a plate. And so that's exactly what was done in this particular case. A large diameter fat probe was used, large spots, sampling of that slurry as it's mixing in a vial. And so this is a very simple setup done in the laboratory so you could begin your development process. This is where we would take the conversion, and we started with a 50-50 mixture of the kinetic and thermodynamic products and allowed it to mix. We can put a probe in, or you can do this non-contact. We did this non-contact. We eventually went with a probe and put it in and proved that we could use it either way and watch that form conversion from 50%, just over 50% to 100%. So this was a nice example of working with the spectrometer, doing it in different aspects, non-contact and contact with an inline probe to get the same results. Once it's gone into the pilot plant, you can take that exact method that was non-contact and in a laboratory with a probe and transfer that to a large probe in the pilot plant setting. And that's part of the versatility of being able to use a Raman instrument with access to non-contact and various size probes, from a micro probe, a typical 18-inch laboratory probe, to an 8-foot pilot plant probe. And we can transfer the methods from instrument to instrument with the different probes, as long as it's the same type of instrument with the models that we built. And that would be a future discussion or presentation on the type of models we built. We do a lot of ratioing in order to increase that versatility. And you can see in this particular case, we add the N3, just over 20%, that's charged, and we can see when the reaction is completed. So we're very happy. It's a fast reaction once you get that conversion going. With this went in a lot of work in terms of, well, how much do you need to put in the N3 form to help kick it over a lot quicker to the thermodynamic form. And so you would spike it with 5%, 10, 20, 30, and so on, and decide what's the minimum to give you quality material in a time frame that you're willing to work on in a pilot plant environment. This particular case is a form conversion, and this is very nice work that was done between Robert Wethman and Ming Wang in the group, and the last topics I've been discussing here were actually developed by them in the laboratory and then taken into plant environment. This particular case, we're going from a monohydrate to a dehydrated form, but we want to keep the monohydrate. The final form should be a monohydrate. So you want to study what are the limitations, what parameters in order to maintain the monohydrate and not kick it over to the dehydrated form. So obviously we're going to run it to the dehydrated and see what those limits are. So there's a number of differences in the spectra that we can utilize. We're going to start right from the beginning. Let's try drying it at 50 degrees. And then the temperature was increased to 70 degrees. And so you're at the monohydrate level at 50 degrees C, and as soon as you increase the temperature, you drop or remove that monohydrate, and it becomes dehydrated. So we want to avoid that dehydration. So let's take this a little bit further. How about a more in-depth temperature study? So what Bob and Ming did was increase the temperature 30, 35, 40, and walk that up and see what is the upper temperature limit. You want to be able to dry the stuff quickly and efficiently, but you want to maintain that monohydrate. So in this particular case, you can see very nice the graphic results from monitoring the changes in the spectra. Now, if we add a nitrogen purge, that's going to help remove the water, obviously the solvent that you're trying to remove, but you're going to remove that layer of water depending on the temperature and, of course, the purging of the system with nitrogen. So again, temperature study here, and you can see if you're going to approach 25, 30 degrees with a strong nitrogen purge, you're going to dehydrate your material at a lower temperature. So it's something that you take with a grain of salt. Now, do you want to run at a low temperature with that purge, but it's still going to take you a very long time to do that conversion. Now, we did this on a very small lab scale in a particular type of dryer, and what Bob and Ming did was look at the drying mechanism and determine that if we were to purge it at an elevated temperature, dry it under vacuum with an elevated temperature and moist nitrogen, we can help maintain that monohydrate and prevent it from drying. So this is what was decided upon. You can run this up to 60 degrees under vacuum. If you're going to use nitrogen, purge it with wet nitrogen, and that'll help maintain that monohydrate, and you can run that at a lower temperature. Again, improving efficiency while maintaining quality material, in this case, the monohydrate. And the last thing I'm going to touch on here is an FTIR method that was developed for product stability. This is a combination product where we're adding a polymeric ingredient during the formulation. And there's also an acetate present, and we need to maintain that. So we're looking at the stability of maintaining the acetate and not having any decomposition. Here we could see the acetate again, plus the active pharmaceutical ingredient and the polymer. Now, in some cases when something fluoresces, we have to avoid Raman at our typical 785 nanometers. So in this case, we ended up going with the FTIR, so we have a nice acetate peak, but we know that the API and the polymer is really coalescing in the same spectral region. But we are focusing on that acetate. As you can see here, after in the drying chamber, and so we're doing small-scale development work, we're looking at the moisture content, we're analyzing what acetate is there, and at a particular temperature for a period of time, 40 degrees, 75% relative humidity, we ended up losing that acetate. So that's going to give us an indication of what kind of conditions we need to maintain. So you want to build a model off of this. And this just gives you a quick rundown, a quick example of using multiple experiments in order to build a model so that we can generate one to apply both in laboratory studies and then in a production environment. And so we have very good agreement between individual results and our average results used in this drying protocol while trying to maintain that acetate. You can see here our drug product stability then went into some additional work to look at being closed up at various temperatures. So they'll take material and seal it up in the bottle and then expose that bottle to different conditions, 30 degrees C, 50 degrees C, over a period of time to look at stability. And again, both temperature and humidity can cause sample degradation. And the IR was used as the primary tool for this drug product stability study. And obviously, we want to look at other methods. So we also look at solid-state NMR, which takes quite a bit of time to run. We also use 2D NMR. Again, that takes time and sample preparation in both cases. But with infrared, it's a very simple experiment to run, to take your material and analyze it by FTIR and look at the amount of acetate that's remaining and the stability of that drug product in this mixture. So in summary, we use both FTIR and Raman spectroscopy to monitor physical changes and chemical changes. And I've also added in here reagents and why we actually use these techniques. I also want to point out that all of this work has been presented at SciAx 2016 and 2018, and some of it will be presented at SciAx 2019 occurring this fall. I'm going to hold off here and pass the baton over to Karen and Stephen. Thank you.
Thank you, Dr. Wasylyk, for your presentation. Before I introduce the utility of Raman spectroscopy in continuous processing of liquids and solids, presented by our next presenter, Dr. Esmonde-White, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the presentation. So please allow me to now introduce our second speaker. Karen Esmonde-White is a senior Marcom specialist at Kaiser Optical Systems, Inc. She completed her PhD in biomedical engineering at the University of Michigan in 2009 in Professor Michael Morris's laboratory and has an MEng in pharmaceutical engineering and an MS and BS in chemistry. Karen is also an adjunct faculty researcher at the University of Michigan Medical School, where she works to develop Raman spectroscopy for new biological and clinical applications. In addition to her research, Karen is an active volunteer in the analytical chemistry community, including her volunteer work for the SciAx Conference, Society for Applied Spectroscopy, and the Koblenz Society. She serves as an ad hoc reviewer for spectroscopy, clinical, and biomedical optical journals, was program chair for the 2018 SciAx Conference, and continues to serve as a biomedical program co-chair for SciAx. I will now be handing over to our second presenter. Welcome, Dr. Esmonde-White.
Thank you, Stephen, for that kind introduction, and welcome everybody to this webinar. It's a pleasure to be here today. I'm going to focus on continuous manufacturing and how Raman spectroscopy as a PAT has a role in this. We know that continuous manufacturing approaches have been used by chemical and oil and gas industries for many years, and this concept is now taking traction in pharmaceutical and biopharmaceutical industries. And why is that? It's because you can make a better product more quickly. You can start to incorporate process automation concepts. It reduces the environmental and safety risks. It also, importantly, redefines what is a plant environment. So we see here this picture, this is from the MIT Novartis Center for Continuous Manufacturing. And this setup allowed product to be made in 14 operations in 47 hours versus the traditional batch process, which involved 21 operations over 300 hours. And continuous manufacturing typically involves process intensification conditions. And because of this, inline PAT becomes more important because you don't want to pull samples that could pose a safety risk to your staff, or you don't want to wait for results. And so I'm going to focus on how does Raman spectroscopy fit into these continuous and batch processes, because it provides specific and quantitative compositional analysis, and the fiber optic sampling probes enables remote sampling. Raman spectroscopy is one of PAT's analysis used in the industry. It does have several advantages. So we can see here, for example, a comparison of Raman spectroscopy of polyethylene versus near-infrared. The bands in the Raman spectra are very distinct, and the spectra are comparing polyethylene at various densities. The envelope between 1400 and 1500 wave numbers shows very distinct changes, whereas those differences are more subtle in near-infrared. And that specificity that Raman spectroscopy has enables method development as well as method transferability without significant method rework. Raman spectroscopy is also compatible with classified environments in an ATEX or North America certified configuration, and it's also compatible with process control software. The sampling flexibility enables analysis pretty much anywhere, from slurries and feeds, turbulent mixtures, gases, biological tissues, and aqueous systems and solids. And so as John had mentioned early in his talk, the Raman spectra from a sample or process can provide the qualitative information, so it can specify what you're looking at. And we see here an example of 3 different polymorphs. It can provide quantification. The center spectra here are showing quantification of glucose in a mixture. And really important in process monitoring and control is, has it changed? So looking at the Raman spectra over a time course, and we see that over on the far slides here. So to kind of take a step back and see the history of Raman spectroscopy and how far we've come even in a short time period, go way back to the inception and discovery of the Raman effect in 1928. So we can see Raman spectra photographed in 1928, used, a sampling configuration that was very similar to what was used in the 1990s. So you have some freestanding optics. Later on, we had incorporation into a microscope. But it wasn't until the late 1990s that we had development of the first generation of fiber optic probes for industrial applications. It was recognized very early that Raman can directly measure in reactive chemistries, and so in-process reaction monitoring was one of the first applications of industrial Raman spectroscopy. These principles of using Raman can be easily applied to flow chemistries, where there are often more intense reaction conditions, and into continuous solid measurement. And since the late 1990s, we can now start to get representative sampling in solids and turbid media by changing the geometry of the collection fibers with respect to the excitation. So this is an example here using a concept called spatially offset Raman spectroscopy, where you're physically separating the excitation from the collection fibers. And we can see that, that D1, D2, and D3, it samples different volumes. And so nowadays we have a lot of different options within the academic laboratory all the way up to process, industrial applications, where we can either harness the effect of optical scattering or minimize the effect of optical scattering depending on the process. So what's typically used is backscattered Raman, where the illumination and collection fibers are relatively close to each other, and that's used in liquids process analysis, as well as if you want to get a surface area of a solid. In order to get a more 3D volume type of measurement, you would use what's large volume Raman, and I'll go into this in more detail later on as well. I previously mentioned swords in my last slide. A variant on the swords is transmission Raman, where you're using a defocus laser to go through the entire sample, and then you collect this, the signal on the other side of your sample. Some interesting work that we had done at the University of Michigan was to develop Raman tomography, which was a multi-point 3D volume, and we had the application for measuring subsurface volume. Today I'm going to focus on backscattered Raman and large volume Raman because those can be incorporated in line and in process. So this large volume Raman is incorporated into a process probe that we call the FAT technology. Excuse me. It provides representative sampling in solids or turbid media at low laser power, and it's offset, so you don't need to touch the sample. We can see that, starting here on the left-hand side, working from first principles, that we have a defocused laser and multiple collection fibers. The next is a picture of what the probe looks like, and the next picture is that it provides a 6 millimeter spot size at a 25 centimeter offset. And finally, this oval shape kind of shows that the differences of sampling volume that can be achieved using backscattered, which it shows up really small on the surface here, versus this larger cone shape that can be achieved using the FAT technology. So let's look at some examples of continuous liquids process monitoring. And I'm deliberately showing some examples that are 20 years old to show that for Raman spectroscopy, the idea of inline PAT for continuous processes are not new. This is something that has been one of the first applications, and since then the technology has gotten better. In this example, we are using a non-contact optic, looking through a glass reaction chamber to measure a microwave reactor. And this is an example of what the reaction looks like. So again, the spectra on the right-hand side are showing the changes over time. We can see the formation of the intermediate ester as well as the product ester. And based on the Raman spectra, we're able to demonstrate that the intermediate A pathway was the reaction pathway. And so this brought about more process understanding, and then from there you can develop a design space and optimize the reaction. This is another example of the Diels-Alder reaction for endpoint determination and reaction optimization. So again, this is an A microwave reactor with sealed glass reaction vials, and we're using a stacked non-invasive fiber optic probe. And so we were using this to monitor the reactant, intermediate, and product formation at different temperatures so that we could optimize the reaction. So you can see that this is a relatively fast reaction. So we were able to move from a 6-second reaction time down to 10 minutes, and we're able to accurately monitor the product formation. And this is another interesting historical example. You can see that the reference is from 1997, and this is the original paper talks about this boiling, toxic, corrosive reaction mixture, and you really don't want your staff to be exposed to that type of environment. You also want to make sure that your heat generation is controlled. And so for that, you want to control the amount of phosphorus pentachloride. And they started off with batch sampling and offline analysis, and eventually tried FT Raman via a site window. But the in-process implementation required a recirculation loop, and that site glass still needed to be cleaned due to opaque impurity buildup. The analysis time was about 5 minutes, and it required 500 milliwatts to 2 watts, which meant sample heating and window. Moving to Dispersive Raman, they got an in-situ measurement in 30 seconds using 40 milliwatts of power. And the sample heating generated by that 40 milliwatts had no effect on sample selection. So these are, the examples that I've shown are, large scale, some older examples and older reactions, just to give a historical precedent for what's going on now. And this is some new examples of using these smaller reaction vessels. So this is an example in one of the Kone plates. And we see here on the bottom right, that it's an improvised Raman flow cell. So we're seeing that integration of Raman spectroscopy into smaller reaction vessels, flow cells. This is an overview of the explosive formation during continuous production. So again, they're using the Corning Advanced Flow Reactor, as well as 2 continuous stir tank reactors. And so this is where Raman comes in. So after the automated dose of the reagent, the Raman spectra were used to track the reagent and the product formation. And so the take-home from the liquids examples are in-process Raman of reactive chemistries is an established technology. It's been around since the late 1990s. It eliminates the need for a flow cell or recirculation loop. And we have integrated into process control systems, which then enables the feedback-based control, as well as increases your process understanding, your efficiency and safety and quality. And so now we'll move on to some examples in continuous solids process monitoring. So again, just as a refresher, this is the FAT technology from first principles. Again, showing that we have a C sampled volume, and that enables the surface and subsurface to be examined at the same time. And this comes in important when we are looking at heterogeneous solids such as products or drug polymer extrusion. So this is an example of a continuous manufacturing for API-based products presented by the in 2012, where you're incorporating product flows, real-time release, feedback loops, and the idea is that you have the processing time in less than. This is an example at the CSOPS facility at Rutgers University. So on the right-hand side, we see the schematic, and then the photo shows how it's implemented at the university. And recently, Professor Hausner had given a webinar on using Raman chloride, monitoring the blends before tableting for uniformity. So another example is continuous tablet coating operations. And so the coating process during a continuous process is intensified because you have high heat transfer, you have frequent presentation of coating mixture, and again, inline measurements are necessary so that you're not stopping production. This is an example of how Raman spectroscopy can be incorporated into a twin coater for continuous operation. At the top, we see the automated tablet feed hopper, the inline Raman, the tablet cascade where it becomes coated, and then the tablet discharge hopper. So all of this can be incorporated without needing to change the hardware. And we can see that the Raman-based predictions match with the offline weight. And so today, Raman spectroscopy is everywhere in the pharmaceutical industry, from raw materials analysis all the way to GMP in process and real-time release. But looking ahead, how do we incorporate the principles that we've developed over the past 30 years into an instrument or field unit that is ready for a non-spectroscopy? And really, the hardware in Kaiser is a small portion of it. So it's including automation aspects such as status and alarms, automated diagnostics, visualization for a variety of users, including field operators, as well as how it integrates into the process adaptation, which includes the design of the probe, taking into consideration the environment in which it will be run, as well as the compliance. And as John had also concluded, new technologies in Raman spectroscopy are enabling safe, efficacious medicine, more environmentally friendly, safer, efficient manufacturing processes. And what that means to the consumer is a higher quality and at cost savings. And with that, I thank our customers and our partner, and I thank you for your attention.
Thank you, Dr. Esmonde-White. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions via the questions tab located directly below your webinar screen. So now our first question will be, what is your standard workflow when creating a spectroscopy-based method for form control? I believe that's for you, Dr. Wasylyk.
Yes, hello. Our basic workflow is where we're going to start when a chemist or an engineer comes in with samples or questions. Can we monitor the forms that we're trying to make or forms that we're trying to avoid? First of all, we're going to want as many forms that are available. Obviously, quantities early in development are going to be limited. We're going to get as many of the different forms that are available that have been characterized. And then we will collect spectra on whether it's an FTIR, near-IR, Raman, any one of our tools, and to see which one works the best. Now, we do have preferences towards using certain instruments because of scalability, transferability, but sometimes it doesn't work out that all instruments or all vibrational spectroscopy techniques will work out quite well. So you do have to be selective. So we pre-screen. Basically, pre-screen is the name of the game, and then we'll decide which one to do. Once you decide on an instrument or two instruments, you can always develop two instruments or two different types of vibrational techniques in order to look at form conversion, and form stability. Then we will get a hold of enough of those samples and then start to do the conversion online or offline during the process, and then build models in that. But remember, our primary technique, whether it's an HPLC or an X-ray or solid-state NMR, that's our primary technique. We need to have those solid values, and hopefully we can match them with a vibrational technique. And of course, as I mentioned, vibrational technique, it's quicker, it's easier, it's more amenable, And if, some sites we send to don't have all the elaborate capabilities because some of the instruments like solid-state NMR is quite expensive. So that's going to be our general workflow. Get as many samples as possible, evaluate which one of those instruments, and making sure that we get the sensitivity that they want.
Thank you. Our next question is, when considering how to control a process, what technology is your go-to? And again, that's for you, Dr. Wasylyk.
We will, in many cases, use Raman as a first start. We will look for fluorescence, and if there's an issue, then we're going to have to juggle from there and try to find something else. But you can do form differences. We could go into low-frequency Raman. We gave a webinar with you a couple of months ago. People are welcome to look back at that on low-frequency Raman, and with that, we're able to look at forms in a spectral region where it works out in many cases quite well, because we can also look at amorphous material. So that tends to become our go-to. Plus, we have a plethora of probes available, and the instruments also are very versatile. They're very robust. We happen to be very selective on the type of instruments we buy, the vendors, so that we have enough support and availability so that we can run at various volumes, various sizes, and, as I had pointed out, ATEX and UL-rated instruments.
Thank you. Our next question is, would I need to have redundant systems for a continuous process? And that's for you, Dr. Esmonde-White.
I think that when you're in process development, then you'll need to look at what the analyzer and the probe are measuring, as well as the criticality to the overall process. So you want to make sure that if you have something that needs to be measured and it's a critical component, and then yes, you would want to incorporate that redundancy, certainly in GMP. So you can measure using one probe in process development and a pilot, and then start to roll out the redundant systems for GMP.
Thank you. Our next question is, what are the challenges you face when creating a chemometric model? And that's for you, Dr. Wasylyk.
What challenges? First of all, our general rule of thumb is make the chemometric model as simple as possible. I mentioned just briefly in passing, we tend to do a lot of ratios. That can eliminate a lot of headaches, and they're easy to understand and easy to justify when you have to go to the filing status, whether it's FDA or EMA. We'll do derivatives and so on depending on the spectroscopy technique and how much it is needed. We'll do PLS modeling. But we try to keep those models as simple as possible and not elaborate, because, again, you do have to justify it. But remember, the fundamental beginning is your primary technique with a particular sample. That's going to be critical, because all at best we could do is match your primary technique. Now, your primary technique can be HPLC or GC or solid-state NMR. It could also be Class A volumetric pipettes and a certified analytical balance can be your primary technique. But we're only going to match that. And so those are some of the challenges that you're faced with before you begin to collect the data, making sure you have quality data. And then general rule, again, keeping it simple, do ratios. This way, if there's a variation in concentration, in other words, you may be running 50 grams per liter, and the next time it's 51.3 grams per liter, and the next run is 49.8 grams per liter. If we normalize that to the solvent and do a ratio, that helps in that variability when you do a transfer, because you're never going to be spot on every time you prep a large batch.
Thank you. Our next question is, how would a software update affect an inline PAT? And that, again, is for you, Dr. Wasylyk.
We tend to keep the same version would be the best way to test, to say. Now, there are times where if there's going to be a software upgrade, we need to look at both methods, both upgrade and the original data, to make sure that there's no perturbations. But we normally just stick with simple ratios, simple models, that when the software is updated, we want to make sure that the parameters for data collection are the same, intensity is the same, everything is the same. Then we look at the models and make sure that they behave with the new software. So you do have to go through a full validation process, but we tend not to change software, and we've been fortunate enough to work with vendors that have kept the software constant and not made any major changes so that we can continue to run our models, just because there's a lot of back-end work that you have to do if you were to do some upgrade.
Thank you for that. Moving on, with the amount of data generated by these online analyses, how do you keep up with the data storage and making sure that data can be read if a software becomes obsolete? Again, Dr. Wasylyk.
In general, all of our data, we always keep an SPC file. So in other words, many of the instruments will have their own native software version. SPC, we feel, is a fairly generic, general format, so we can go to a number of software packages and open up the SPC files. That goes a long ways for data integrity. I will add, based on the last question, is that if there is a major change in software, we can keep the original software for processing, so that 3-4 years down the road, if we're ever audited and they say, can you show us how this data was processed, we have that original data and we have that original software so that we could show how it was processed in those results. Same thing applies here. In this particular case, we want lockdown, and then we have that original software is traceable. And that could be future talks on what type of software packages we use in order to be Part 11 compliant and traceable.
Thank you for that. Our next question is regarding the PHAT probe. How can you detect and avoid probe head fouling? Now, would Dr. Esmonde-White like to answer that first?
Okay. I should first point out that because the fat probe is a non-contact measurement, it can be used through sight glass, and it does have that 25 centimeter offset. However, when you're working in a spray coat situation, there are some residual particles that can get in. We also do have an add-on, which I guess it's a sheet that you convert the fat probe, which will protect it somewhat from the particles. Some things that you can use to look for are decreases in the overall Raman intensity. If you're seeing a new band, for example, that could indicate probe fouling as well.
Yep. And Dr. Wasylyk, do you have anything you'd like to add?
Well, actually, there's one point I'll just bring up. If I understand for fat probe is non-contact, and that's perfect. If we do have to use a regular probe in a reactor, we're careful about where it's put in the reactor to avoid potential fouling or minimize that. And that would be putting it near the agitator blade so that there's no eddies or small turbulent, semi-turbulent areas, so it would allow for potential coatings. So we tend to put the probe tip near the agitator where there's a maximum amount of shear, so that the probe has the greatest propensity or ability to be cleaned, wiped clean during the process.
And thank you for that. Our last question, so unfortunately all we have time for, last question is, can the fat probe be used for biotech liquid applications? Again, Dr. Esmonde-White, would you like to answer this first?
We have probes that are specific for upstream bioprocess, and so I would recommend looking at those probes first instead of the fat probe. I mean, what you're seeing in upstream is, the increase of particle scattering because you have your, increase of number of cells, and so that optical scattering is varying with time, and that's exaggerated because in the CHO cell example, you're culturing for anywhere between 11 and 17 days. And so we've found what works best is using a probe with a backscattered optical geometry to minimize the effects of optical scattering as trying to harness.
And Dr. Wasylyk, do you have anything you'd like to add?
No, I think Karen summed it up.
Okay. Well, thank you. And with that, audience members, I thank you for your questions. I will finish the question and answer session. Before I conclude the webinar, however, I would like to ask our presenters, Dr. Wasylyk and Dr. Esmonde-White, if you have any closing remarks or anything you'd like to let our audience members know about. Dr. Wasylyk, would you like to start first?
No, I would say that if you do have additional questions, please feel free to contact me. Obviously, my name's available on LinkedIn and so on. As I mentioned, a lot of this data and future data and a lot of related talks are always popping up at the SciEx conference. And I will put a plug in for that since I will be there giving another talk. And I'm sure Karen will be there also. Karen?
Yeah, so I give a similar plug for the SciEx 2019 conference. It's not too late to submit an abstract for a poster presentation. I will be presenting on this topic as well and in upstream bioprocess monitoring using Raman. If you have any questions, like John, I'm on LinkedIn. You can also look me up on Google. Thank you very much.
And I would like to take this opportunity to thank our presenters once again. That was John Wesley, Senior Principal Scientist at Bristol-Myers Squibb, and Karen Esmonde-White, Senior Marcom Specialist at Kaiser Optical Systems, Inc., for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send me over your questions to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.